All right. Well, thank you all for coming. Today, we're going to be talking about the basics of DNA testing for genealogists. And there's a lot of different types of DNA testing options, and they vary in terms of what they can tell you. So I feel that it's important to start out just with a little bit of basic biology. So don't worry, I'm not going to get too sciencey, but I do want to talk just briefly first about the cell. Now, there's a lot of stuff floating around in the cell, which that's actually the technical term for it is stuff. Make sure you write that down. So, some of that is the nucleus. Now, the nucleus contains the majority of our DNA. It contains our chromosomes. Humans have um, uh, 46 pairs of chromosomes or 23 pairs of chromosomes. 22 of those are called autosomal DNA. The 23rd are called the sex chromosomes. They're what determines a person's biological gender. Then you also have mitochondria. Mitochondria is referred to as the powerhouse of the cell. It actually came along later on in cell evolution. It was thought to be a bacteria that came and created a symbiotic relationship with the cell. The cell provided it a protected environment to flourish in, and the mitochondria in turn allowed the cell to metabolize oxygen, which then helped it to multiply and make all of the multicellular organisms that we see today. So because it came along later on, it has its own type of DNA, which we call mitochondrial DNA or mtDNA. So this is great. We have these sex chromosomes, autosomal DNA, mitochondria. Where does it all come from? Well, modern science definitively tells us that it comes from aliens. I'm just joking. It actually comes from our parents, obviously. From our mom, we get half of our autosomal DNA, and we get an X chromosome, and we also get the mitochondria. Now, everyone has mitochondria, both men and women, but we only inherit it from our mothers because it's found in the egg, not the sperm. So the mitochondria that everyone gets passed on from their maternal line, not their paternal line. Then we also get that X chromosome. From dad, we get an half of our autosomal DNA. And if we are a boy, we get a Y chromosome. If we're a girl, we get an X chromosome. So now we know where it's from. But what does that even mean? Well, it means that we have essentially three types of DNA. So we have three test types. We have Y DNA, and that looks specifically at the Y chromosome, which only males have. We have mitochondrial DNA, which everyone has, but they get from their mothers. And we have autosomal DNA, and that does include the X chromosome. Sometimes people confuse the X chromosome with mitochondria, but they're two different things. And these are going to be the three test types that we talk about today. So first, we're going to talk about autosomal DNA. Now, this DNA, as you recall, is a 50-50 mix from each of our parents. So they get it from their parents who get it from their parents. So we get some of our autosomal DNA from all four of our grandparents, all eight of our great grandparents, and so on. And so because of that, it's able to trace all of those different ancestral lines and provide a percentage breakdown of the regions of the world where your ancestors came from. So this is where you find out things like that you're 50% Jewish, or 50% English, or however that breakdown winds up being. And it also provides a list of matches. These are other people who have tested in the database that you have tested with. So if you've tested with FamilyTreeDNA, it's going to be other people who have tested with FamilyTreeDNA. And it's going to be people who share common ancestors within about the last 5 or 6 generations. So why 5 or 6? Why is that the cutoff date? Well, if you think about it, let's say here's you. So you have 100% of your DNA. Hopefully, you have 100% of your DNA. You get 50% from your parents, like I mentioned. It means you get about 25% from your grandparents, about 12% from your great grandparents, then about 6%, then 3%. So anything further back than that, It's probably going to be such a small percentage that we're not going to be able to reliably say that it comes from one ancestor or another. They're going to be such small fragments of DNA that they might appear to be the same in different ancestors. So that's why we say about 5 or 6 generations. But this sort of DNA isn't exactly the 50-50 split that I'm describing here. It goes through a process that we call random recombination. So, an analogy I like to use for that is say that your mom is a deck of cards and your dad is a deck of cards. You shuffle those two decks together, you take half the deck. That's what you get. You shuffle those two decks together again, you take another half of the deck, that's what your sibling gets. So you're working off of the same two decks, but every shuffle is going to be slightly different. So you're going to get some cards in your deck from your mom that your brother didn't get. And your brother is going to get some cards from your dad that you didn't get, and so on. So this 50-50 split, when it comes here to this generation of 25, it really could be more like 20% and 30%, or 15% and 35%. So it can vary a little bit. So that's why we say approximately 5 or 6 generations, because it could be that some of those DNA cards got carried down one generation after another, and the common ancestor that you have with another person, the common DNA that you share with another person, actually came from an ancestor who was 7, 8, 9 generations ago. It could also be that you do have another third cousin, let's say, who doesn't share any DNA with you because they didn't happen to get the same cards shuffled into their deck. Any DNA that you potentially would have shared in common got shuffled out somewhere along those lines in those generations. So it's not quite as cut and dried as this. To illustrate the random recombination nature of it, here is an example of the percentage breakdown that I mentioned. So you can see here that it breaks it down by region. So it doesn't say what country it is. And for the most part, sometimes it does, like with Ireland. Um, but Scandinavia or Central Europe, it's by region. And we'll explain why in a little bit. But these are my results. These are my full-blooded brother's results. His last name was Brusala. He married a woman whose last name was Casala, and they decided to hyphenate their name for Brusala, which part of me is like, okay, well, that's kind of unique. That's cool. But the genealogist in me is like, people are going to be like generations from now. They're going to be like, I'm trying to trace my Brusala line and I can't get past, 2016. What happened? But you can see that his percentages are a little bit different than mine. They're mostly the same, but they are broken down a little bit differently. This one is my full-blooded sister, which again is a little bit different. So you can see that we're all working off of the same decks here, the same parents, but we ended up getting a slightly different shuffle. One thing that I think is interesting with this is that you can see that Mark and Valerie are much more similar in their DNA makeup than either of them are to me, which is interesting because when it comes to personality, temperament, interest, the two of them are much more similar to each other than either of them are to me, which again, I find interesting. Now, these percentages that we get, how do we determine them? Well, we use something called reference populations. So think of a map of the world. Oh, wait, sorry. No, this is from Lord of the Rings. Never mind. Never mind. Wrong thing. Okay, here we go. So and I'm just going to focus here on Europe because that's where a lot of my ancestry comes from. So that's what I'm familiar with. But if you look at this area here, you can see that historically there aren't really country borders. Countries change a lot over time. Country borders do. The country borders that were in Europe 100 years ago are pretty different than what they are today. But even though those country borders have changed, the people who are living there have remained pretty much the same. So what we do are we take those people who have lived in the same area for generations, they've lived there for hundreds of years, and we count those as what we call a reference population. We take their DNA We try to find the unique DNA signature, it's called, or the unique markers that they have that aren't shared by other people. That means that it represents special mutations or changes in their DNA that occurred in that population that didn't occur in other populations. So, for example, if two different groups of people split apart in the Middle East and one went down to the Mediterranean, another went up to Northern Europe, they started developing their own specific mutations, they're going to have some changes. They're going to have some similarities too, but they're going to have differences. And so what we try to do are pick out those differences that are unique to that area. And then whenever someone comes along and tests, you or me, anyone else, we take our DNA and we try to find some of those markers that match up with those reference populations. And then that's what provides those percentages. Now, I mentioned that the markers are unique, but they also share some similarities because, again, in my example, even though they've lived there for hundreds of years, they came from somewhere else. So we like to think of the DNA for these reference populations as being completely distinct. For example, here's Scandinavia over here. It doesn't have anything in common with Western or Central Europe, which doesn't have anything in common with the British Isles. But that's not really the case because we're all human. So we all share a lot of our DNA in common with each other, regardless of where we live. So really the reference populations look a bit more like that. There's differences, but then there's similarities. Now, as time goes on, if you've done a test, you've probably noticed over the years you've gotten an email from one of the testing company that you've tested with saying, hey, we've updated your ancestry profile or your results. And that's because we've been able to get a better and better idea as more people test of what actually is unique. If you have a small reference population, you might say, oh, well, these people don't have this marker, these people don't have this marker, therefore they're totally separate. But maybe some other people in those populations do have a little bit of overlap. So as more people test, as the science improves, that overlap gets less and less. So you get more refined results. But again, I talk to a lot of people who say things along the lines of, um, I talked to one person, they said that they were Irish. They knew they were Irish. They were 100% Irish. All of their ancestors were Irish, but the test was coming back and it was telling them that they were only 98% Irish and 2% from Western and Central Europe. And that must be wrong. Well, if you think about it, Ireland and the coast of France are pretty close to each other. So historically, they've had a lot of interchange between those two groups of people. So genetically, they're more similar to each other than, say, someone from Ireland and someone from Southern Africa would be. So to illustrate the differences that you might see from one testing company to another, because each testing company has their own reference populations. This is a breakdown of my DNA as seen by the three testing companies that I've tested with, FamilyTreeDNA, 23andMe, and AncestryDNA. So you can see here that it's a little bit different. We have England, Wales, and Scotland over here. Over here, it's described as British and Irish. And then the same thing here, we have Northwestern Europe as opposed to, say, Scandinavia. It essentially references the same thing, but it has some slight differences in those populations. To give you an example of how radically it can change over time, a common question that we get a lot are people who have Native American ancestry or who they have a family story that they've been told that they are Native American. Let's say, as an example, that they have a Cherokee ancestor. So they take a test and it doesn't come back as Native American. So does that mean that they don't have Native American DNA? Well, no, it's not telling you that you don't. So let's explore some of the reasons why that might be. Well, first, let's talk about the reference population. When FamilyTreeDNA first did this test, the reference population that they used for Native American was an Amazonian tribe in South America. because it was a very isolated group of people that hadn't had contact with other people in that area, Europeans or other settlers. Because when you think about the history of North and South America and the colonization of that, it's very hard to find a group of people that haven't been anglicized in some way, that they haven't essentially intermixed with different European groups. If you have a group of people who have mixed heritage, it's not ideal to use them as a reference population because you don't know genetically which markers they got from their European ancestors and which ones they got from their Native American ancestors. So we chose an Amazonian tribe because it was very isolated. But if you have an ancestor who you believe is Cherokee, well, that eastern coast of North America and the Amazon rainforest, while they're both in the Americas, they're pretty geographically different. So they've split apart. They split apart from the rest of the world, from Asia and Europe, thousands of years ago. The estimate is somewhere between 14 and 30,000 years ago. So that's a lot of time for them to be unique. But the group in Eastern Europe split apart from the group in the Amazon still several thousand years ago. And that's enough time to develop differences. So if the Native American markers that you have from your Cherokee ancestor weren't shared by any of the people in the Amazon, then it's not going to come back as Native American because those markers that are in the reference population aren't present. Now, this has improved over the years. We now have a North American reference population, which are the Havasupai, who live in the bottom of the Grand Canyon in Arizona. Again, they're a very isolated group of people. But still, if you're talking about the Southwest United States versus the eastern coast of the United States, there's going to be some difference. So, what I always like to tell people is that the absence of evidence is not the evidence of absence. Just because it's not coming up that you have Native American ancestry doesn't mean that you don't have Native American ancestry. A few other reasons why it might not show up, again, is that random recombination that we talked about. It could be that even if you are of Havasupai descent, if your Havasupai ancestor was more than 5 or 6 generations ago, it could be that those Native American markers didn't make it through those shuffles down to you. So that's another reason that you might not see it. So these are just some things to think about. When you get a test, I always tell people to take these with a grain of salt. So next, I want to talk about the next aspect of our test, because regardless of the testing company that you go with, you have, with autosomal DNA, you have two main aspects. You have the ethnicity report or the regional ancestry report. They call it different things in different places, but essentially it's this breakdown that we've talked about here. And then you also have matches. Now, these again are living people today who have tested at your testing company and share DNA in common with you. And this is sort of the bread and butter for a lot of genealogists because they're people who are trying to make connections to find other relatives who may be able to help get them past a brick wall that they have in their family history. So we're going to talk about matches. Now, matching is optional because there are some people who are only interested in that ethnicity report and they don't want to have their information out there. They don't want to have other people seeing their name or their email address or anything like that. It's optional. You can opt out of matching so that you don't see other people in the database and so that other people don't see you. But of course, if you do that, it means you can't play with matches. And, where's the fun in that? So I want to talk a little bit here about the matches. So this is an example of, this is one of my cousins who have tested, one of my first cousins on my mother's side. So you can see here that this person, they match my two siblings, and then this is another one of my cousins who has tested. And you see that there's a bunch of information here. Up here you have ancestral surnames. So these are people who I have added to my tree. And this information is there so that if you see me as a match, you can take a look at my tree or my list of ancestral surnames to be able to see if there's any names that you see in common. That can give you a hint of, oh, if you have, say, a Scarborough in your tree and you see that I've listed Scarborough, that's an indication that that might be how we connect. You can also then see down here that it says shared DNA, and this it says CM. CM stands for centimorgans, which is a unit of measurement for DNA. So the more centimorgans that you share in common with another person, the closer that match is going to be to you, because there's been fewer shuffles that have occurred um, back to your common ancestor. So if you have more in common, then that person is more likely to be, say, a first cousin in this case than they would a second cousin or a third cousin or something like that. It does say over here that there's a relationship range. So it doesn't spell out that this is my first cousin, or, well, I should say this, it's, I am my first cousin. We're looking at my cousin's report again, remember? But it doesn't come out and say first cousin. It does down here because I've attached them to my tree, but the relationship range that we see up here of uncle, aunt, niece, nephew, grandparent, grandchild, or first cousin is because all of those relationships share potentially the same range of centimorgans. Because again, there's that shuffle. So you have sort of a minimum amount that you would share with another person and a maximum amount. You also see here that you have a longest block. Now, if we go back to our deck of cards analogy, where your mom is a deck of cards, your dad's a deck of cards, you shuffle them together. Let's say that those are brand new decks. You've just unwrapped them, you just opened them, they haven't been shuffled, so it's just you have an ace of spades, two of spades, three of spades, four of spades, five of spades, all that sort of thing. If you only shuffle those two decks together once, you're probably still going to have a lot of chunks in common that are, that three of spades, four of spades, five of spades that haven't been shuffled. And we refer to those unbroken segments of DNA as blocks. So the longer the longest block, the potentially closer that relationship is to you. So it could be that you have fewer centimorgans in common with a match, but if you have a longest block with that match, then that person is still probably going to be closer to you. So you can see here, for example, these two people here, these are both first cousins of mine. I have not, I blanked out this name because they chose to keep their information private, and I respect that. But you can see here that they're both first cousins, but they share slightly different amounts of shared DNA and a slightly different longest block. Then lastly, you can see here that this person is an X match. That means they match me on my X chromosome. Which means for me that this would be a person who's on my mother's side, because me as a male, I only have one X chromosome that I got from my mother, not my father. So the fact that it's on my X chromosome means that this person has a common ancestor on my mother's side. There's more in to go with X matching. If I have time at the end, I'll get into that a little bit more, but I do want to cover the other test types as well. So I'm going to go ahead and move along to Y-DNA. So Y-DNA, unfortunately, it is males only. So this means that if you want to get a Y-DNA test and you're a female, you would need to get someone like a brother or your father or an uncle or a first cousin or something like that tested. So for the ladies in the audience, I know that can be a bit of a drag trying to find someone who's like that. But the plus side is that scientists have definitively connected the Y chromosome to the inability to ask for driving directions. So you have that going for you. It's a bit of a little toss up, plus minus there. But the Y DNA, it traces the direct paternal line. So in other words, the father's 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 line. So you don't see that shuffle, that mix up that you get with the other lines. With autosomal DNA, you have all of these different lines who are all contributing. And that's why you wind up with a percentage. In this one, it's different because you're only looking at an unbroken line. I got my Y chromosome from my dad, who got it from his dad, who got it from his mom, his dad, and so on. So this test can go a lot further back. The matches that you can get with this could potentially share common ancestors within 25 generations of you. There are different levels of Y-DNA testing. The way that Y-DNA testing works, it looks at something that are called STR markers. It's a little bit different than the way autosomal works. And with the STR markers, we have different marker levels. We have anywhere from 12 to 111. So you can think about these sort of like they're a puzzle piece. If you have a puzzle that has 111 pieces to it, whatever marker test that you're looking at that number of pieces. So again, as generations go on with this, you're going to get slight mutations. So that doesn't mean that you have some sort of deformity or anything like that. Mutation just means that it's a slight change in a value on your DNA. It means that you got maybe one extra copy of a particular marker or one fewer copy of a particular marker or something like that. It doesn't affect your ability to function or live a normal, healthy life, but those small changes do add up over time. So for example, let's say that you have a father, and we're only looking at those 12 markers. You have a father, and he has 3 sons, and 2 of those sons, 12 out of 12 markers, they're identical. But on that 3rd son, we saw a slight change in one of the markers. Well, that son who has the slight change is then going to pass that slight change on to his descendants. And then if any of those descendants have a slight change, they're going to pass it on to their descendants. So by looking at how many of the markers a person has identical to another person versus how many they have differences gives you an idea of how far back in time that common ancestor has been. Because the further back they are, the more chance you have of having a change on any of those markers. And so we call that distance a genetic distance. So this one doesn't give you a relationship range like an aunt or uncle, niece or nephew. It gives you a genetic distance. So in talking about this puzzle analogy, if you have a puzzle, again, with 111 pieces to it, if you're only looking at 12 pieces, if you match another person 12 out of 12, you might think, oh, well, this is great. We must be really closely related. There probably haven't been a lot of generations for a change to occur. But keep in mind that you're only looking at a small section of that puzzle. You're missing the rest of it. So as you test more markers, as you look at more of the puzzle, you're able to say, let's say when you go up to 25 markers, oh, well, I'm actually missing a few pieces over here. So we're probably not as closely related as we thought. So the more markers that you look at, the more accurate you get an idea of how you match another person. So let's take a look at this in action. So this is a list of my matches here. So again, I've blanked out their first names just for privacy reasons. So you can see here that at 12 markers, I have 375 matches. So that means I have 375 people who share with me 12 out of 12 markers. Well, obviously, some of those people are going to be closer to me than others. So how do I differentiate that? Well, let's take a look here. Don't worry about anything that says big Y. I'm not going to talk about that here, but you can see here that I have an exact match, 12 out of 12. When we go up here to the next marker level, to 25, you can see that it's weeded out over 300 of these people. I now have 36 matches in common with them. And Some of these are going to be closer than others. You can see here that now instead of an exact match, it says one step. So that means this person, Tim Brewster here, who I just arbitrarily named Tim, I share with them 24 out of those 25 markers. And you can see here on the left how many markers they've tested. It does say that it goes up to 700 here. I know I mentioned 111. The extra ones are part of the big Y, which again is not for matching purposes, and we're not going to discuss here. So you can just think about this like it's 111 and they've taken all of them. So now I have these people who are all one step different. Well, again, some of them are going to be closer than others. So when I go up to the next level of the 37 marker level test, I have 34 matches. So I still have a similar amount. Now, before I go up to the next level, keep in mind that some of these people, this one here, they have only tested up to 37 markers. So when the test first came out, when Y-DNA testing first came out in 2000, to be commercially available, the most that was available at that time was 12 markers. And that was considered to be good enough for are you related, are you not? Then as time went on and the cost for testing and the technology improved, we were able to provide more markers at a reasonable cost. So it went up from 12 to 25, and then from 25 to 37, and so on. So depending on when your match has tested and what level of testing they've decided to get, they may not have all 111 of those markers. So these people here that have tested up to 37, when I go up to the next marker level of 67, they're not going to show up on that match list. And that's because I don't have those extra 30 puzzle pieces, or rather, they don't have those extra 30 puzzle pieces for me to be able to compare to. So without that information there, we don't know if they're actually close or not, so they don't appear at the higher levels. So keeping that in mind, when we go up here from 37 markers to 67, it cuts it down to 18. Some of those are going to be because they haven't tested. Some of them are going to be because we have found pieces that aren't missing or that are missing. And you can see that here, up here with Tim Brewster, that now he's gone from one step to two steps. So this again shows that I'm not quite as closely related to him as I thought. I am still pretty close, but not as close. But these other people here, you can see that they have two steps as well, and that some of the names have changed. That's because some of the people have dropped off for whatever reason. When I go up here to the final level of 111, I now have 7 people who've tested. Now, if you remember from the previous slide here, Tim, he's tested 700. That means that he is potentially going to show up on my 111 match. And so far, it seems like he's pretty close. But when I go up to 111, now all of a sudden, my closest match is not Tim Brewster. And Tim, in fact, does not show up on the match list, even though he has it. It means that he has so high of a step difference that he doesn't show up, because each level of testing has a cutoff for the number of steps that are allowed. And that's generally because if you have too high of a ratio of steps missing, then that person is probably related to you so far back in time that you're not going to be able to compare records to find a match. In other words, it's not going to be genealogically useful. And since these match lists are for genealogists, that's why we have that cutoff there. So, for example, at the 37 marker level, we have a cutoff of 4. At the 67 marker, we have a cutoff of 7. 111 marker, we have a cutoff of 10. So this top person here, this is now my closest match. You can see that they have a 4-step difference. But one interesting thing is that as we've gone through here, we've seen a bunch of different surnames. We've down here at the 12 marker level, it's all over the place. Here we start seeing more Brewsters. Here, again, we have a bunch of Brewsters, changes slightly here. But at 111, again, my closest matches are Brewsters. So this is a way that it confirms to me that I haven't had an NPE, we call it, a non-paternal event, or some refer to it as not the parent expected. So if you start seeing names that are different, it's an indication to you that potentially somewhere along the line there was an undocumented adoption, or in some way that the father wasn't actually the father. So you can start to see a bunch of different surnames that are here. One thing that can help you make sense of the surnames that you're seeing are things called group projects. Now, group projects focus on things like surnames. So, for example, I'm part of the Brewster Surname Project. It requires a Y-DNA test to be a member of it, because the Brewster surname is passed down through the paternal line. Y-DNA is passed down through the paternal line. So it's a good indication of being able to tell how close I am to other Brewsters. So if you as well are a Brewster and a test comes back and you're not seeing any Brewsters, you could join the Brewster Project to get an idea of maybe when that NPE occurred or why you're seeing other names. All of those group projects are free to join. There's thousands of them. They're all run by volunteers who are specialists in whatever their topic is. There's other topics as well other than surnames. There's things like geographical regions. If you're interested in your ancestors who are from England, for example, there's an English surname, or I'm sorry, an English DNA project that you can join. Things like that. They require different types of tests depending on the type of project that it is, but they are all free to join. So that's that for Y-DNA matches. Now, one thing that you see here is that it lists a haplogroup. So a haplogroup is essentially a migration route out of Africa. So it's going to look something like this. Now, it's out of Africa because the oldest humans that we see in the fossil record and that lines up with the genetics of the people that we see around the world today, the oldest humans are found in Eastern Africa. And this was hundreds of thousands of years ago. And from there, they spread all across the world. Now, when I was talking in that example of autosomal DNA, how when two different groups of people split apart, they started having specific mutations or specific changes on their markers that occurred in one group of people, but not another. The same thing happens with Y-DNA. And when we trace those differences in Y-DNA, we get a haplogroup, which is a migration route out of Africa. Now, it gives you a direct line migration route because, again, your father came from one specific place. His father came from one specific place. His father came from a specific place. You're not having all of these different ancestors, potentially from totally different parts of the globe, who are all contributing to your DNA. You only have that one paternal line. And if you trace that paternal line back far enough, hundreds of thousands of years, the common ancestor that we all share is in Eastern Africa. And we call that common ancestor Y chromosome Adam, which isn't necessarily the biblical Adam. It's just a representation of the DNA of the oldest humans that we see today. So these haplogroups are signified by a letter. So you might have seen, for example, the RM269 haplogroup, or the R1b haplogroup, as it's sometimes called, or the J haplogroup, or the E haplogroup, things like this. If you've done testing, this may ring a bell for you. These all indicate different groups of people who have split apart and their general migration area. And the things that follow after that are subgroups within them. So the R haplogroup, for example, is found commonly throughout Europe, but there are different branches of the R haplogroup tree that we see in different parts of Europe. And so each one of those are subdivided into things like R1b or R1a. Lastly, we're going to talk about mitochondria. Now, again, mitochondria, everyone has, both mothers and fathers. Everyone has it, but we only inherit it from our mothers. So this test is almost the opposite of Y-DNA, because instead of tracing the direct father's line, it's tracing the direct mother's line. So it's looking specifically at your mother's, 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 and so on. So it's going to tell us roughly the same information that Y-DNA does, only it's going to be telling us about the mother's line. So mtDNA is split up into three different areas. It's split up into the hypervariable region 1, or HVR1, the HVR2, and then the coding region. So you can think about these three areas the same way that you think about the different levels of Y-DNA testing. So with Y-DNA testing, you had things like the 12 marker level or the 25 marker level. With this, it's going to be the same thing for matching, but it's going to be at the HVR1 level or the HVR2 level. And it does the same thing. As you look at more of the DNA, you get a better idea of how close one person might be to you versus another. Now you can see here that it's a bit disproportionate. HVR1 and HVR2 together only make up a little less than a third of mitochondria. The rest is taken up by the coding region. So let's take a look here at the matches. Now these I've blocked out again for privacy reasons. You can see up here that they have the name. If they have listed an earliest known ancestor, it'll show there the same way as you saw surnames with autosomal DNA or an earliest known ancestor with Y-DNA. This is the earliest known ancestor for their mother's line. Now, this I always tell people to take a bit of a grain of salt with, because this is reported by genealogists. And as you may know, not all genealogists are created equal. And sometimes people confuse the maternal side with the direct maternal line. So I have one match who has listed their earliest known ancestor as a John J. Now, I don't want to judge. It's possible that John was the name of their fifth generation great grandmother, let's say, but it's more likely that it was the fifth great grandmother's husband. And that was the earliest one that they knew of on their mother's side. So that's the name that they put. If you've done genealogy, you know it can be incredibly hard to trace the maternal line because sometimes they're only listed as, say, Mrs. James Brewster. So it's hard to trace that particular surname or what the maiden name was for the mother. So that can be the advantage of mtDNA, because even though the surname might change every generation as each new mother takes on the name of their husband, even though that might change, the mtDNA itself doesn't. So this can provide additional clues. Now, the downside of mtDNA matches are that they can potentially be even further back in time. They can be as much as 52 generations removed. I'll say that again, 52. That can be a lot. It is so much further back in time because mitochondria takes a longer time to change. If you remember our diagram of the cell, you only have the one nucleus, but you have a whole bunch of mitochondria that's all floating around in the cell. And sometimes a change will happen in one of those little mitochondria, but not in the other ones. And that little change that gets passed down in the egg is going to take a little while to catch on to the rest of them. It might take a few generations for that change to be reflected in all the mitochondria that's in a cell. And because of that slow process, it means that it's slower to change, it takes more generations, therefore you and another match might share a common ancestor who's further back in time. So one thing that I tell people with mtDNA is that it works really well in tandem with autosomal DNA, and I'll get into that in a bit. But first, I want to talk here briefly just about the matches and what we're seeing. So you can see here that in the haplogroup, again, it's represented by a letter followed by a number. And that's going to be the same thing that we saw with Y-DNA, with our example of R1b, let's say, in this case, it's U5. We have the earliest known ancestor, and then over here we have listed the type of test that they have provided. Now, this is valuable. FMS, it stands for full mitochondrial sequence. It means that they have looked at HVR1, HVR2, and the coding region. Not everyone has, because again, mitochondria has evolved the same way that Y-DNA testing has, where more and more of it has been looked at. First, it was just HVR1, then it was 1 and 2. Now it's the full sequence. This bottom person here, it's a little bit cut off, but you can see they've only taken the HVR2. That means they're not going to match me at that full sequence level. But this is valuable because if I have a person who is a mitochondrial match, it might be that it's far back in time. It's a little bit hard to tell because those mutations don't happen every single generation. So it's hard to gauge accurately how the exact number of generations. But if a person has also taken a family finder test, which is our name for the autosomal DNA test, then you can compare to see if that person is an autosomal match to you as well. And if they are, then we know that they're within 5 or 6 generations. So that's how these tests can work in tandem together. Because if you have someone who's a mitochondrial match and an autosomal match, then you know that they're on the direct maternal line. You don't have to guess which ancestor they came from, and you know it has to be within 5 or 6 generations. So let's take a look at this. If they have this little symbol here highlighted in blue, it means they've uploaded a tree, and then the type of test that they have. So now up here, we're looking at HVR1. You can see here that I have 34 matches. If instead I select HVR1 and 2, that's going to narrow the range down. Now I'm going to see that I have 19 matches. Now it does cut off the people who only had HVR1. And when we go up to the next level, it's going to cut off the people who only have HVR2. But when we go up to the top region, we can see that these are going to be the people who are potentially the closest to me. Now, you may notice here that genetic distance is only shown when we look at the full sequence of it. But again, remember that with mtDNA, HVR1 and 2 take up such a small portion of the entire genome that if you mismatch on even a small part of it, then you're probably going to be too far back in time to really be genealogically related to each other. So that's why we show that. But again, genetic distance means the same thing. It's how many markers you don't share with another person. Lastly, you get a haplogroup, which is the same as the Y-DNA, again, but with the mother's line tracing you back to Africa hundreds of thousands of years ago to mitochondrial Eve. Again, not necessarily the biblical Eve, but the mitochondria that we all share. So to finish that up, a test type comparison. So we have mtDNA, which we just talked about. That one only studies the direct maternal line, provides matches within 52 generations. The matches must be on the maternal line, and it provides that maternal haplogroup. Y-DNA is the opposite. It only studies the direct paternal line, must be within 52 generations, must be on the direct paternal line, and it provides the haplogroup. Autosomal DNA studies all the lines, provides matches in 5 to 6 generations. The matches could be from any line, though. So each of them have some strengths and minuses. With Y-DNA and mtDNA, you know automatically which line it's on, but you don't know how far back it is. Could be pretty far back. With autosomal DNA, you don't know what line it is, but you know it has to be probably fairly close, within 5 or 6 generations. So that's how having more than one test can provide additional information. Now, a question that I get a lot is, well, what test should I get? Or what's the best test? Well, I always tell people that's a bit like asking, what's the best tool? It's like walking into a hardware store and saying, I want you to give me your best tool. Well, the guy at the hardware store is probably going to ask you, well, what are you hoping to do here? Because, I can sell you the best hammer that we have in the store, but if you're trying to saw some boards in half, that hammer probably isn't going to do you a lot of good. And then vice versa. So it really comes down to, what are you trying to get out of testing? What genealogical puzzle are you trying to solve? If you're trying to solve a puzzle on your maternal line, if you're trying to prove that two people share a maternal ancestor, mtDNA is going to be your best bet. Same with Y-DNA. If you're trying to prove a paternal ancestor, go with Y-DNA. If you're trying to prove something that's in between, then it's going to be autosomal DNA. That one's going to be the most that one is the most widespread. That's the one that is provided by all testing companies and has by far the biggest database, because you have a lot of people who are trying to find, say, their great grandmother's father. And every time you skip a generation, if you try to go to your father's mother, whenever you go outside of these two outside brackets, then mtDNA or Y-DNA are no longer helpful for you unless you're able to get a cousin or some other descendant who traces back to this mother's or this father's mother. So if you get a first cousin who shares this maternal grandmother with you, then they would be a person to test for Y-DNA if they're a son of a son. Say your maternal grandmother's brother had a son who had a son, you could get that son tested. Basically, however, you can trace back to the specific ancestor that you have the question on. So, last, I want to talk about the testing options. So, there's a lot of different options out there. The three biggest ones that I'm going to highlight: Ancestry DNA, 23andMe, and ftDNA. Ancestry DNA, they provide the autosomal ethnicity report and matching. They tell you the mtDNA and Y-DNA haplogroup, but they don't do matching for that. They look at a different kind of marker, and they don't compare markers to other people. They look at specific mutations that define some of those major haplogroups, like U, or in my case, U5. They don't, so they provide the haplogroup, but not the matching, but they do also have the largest database by far, hands down. A lot of people have trees on Ancestry as well, so that is a plus to be able to compare people's trees. 23andMe, they again provide an autosomal ethnicity report and matching. They do provide the haplogroups, but not Y-DNA or mtDNA matching. They also provide medical information, so that is a plus for some people, and they have the second largest matching database. FamilyTreeDNA, when it comes to autosomal DNA, has comparatively a small database, but we still do have over, I believe we just topped 2 million people. So it's still a fair size. We do provide autosomal ethnicity reports and matching. It's the only company that provides mtDNA and Y-DNA haplogroups, or I'm sorry, Y-DNA and mtDNA matching. So it has the biggest variety of tests. So it's not necessarily that one company is better than another. They each have their strengths and they each have their weaknesses. Now, one thing that you can do is, if you've tested with another company, you can download a copy of your autosomal results and you can transfer them to ftDNA for free, and you can be added into their autosomal matching database. So this holds true for Ancestry DNA and MyHeritage at this time. We're actually the lab that does the testing for MyHeritage DNA. So those test results are 100% compatible. Right now, the transfers from 23andMe have been paused. You may have heard a few months back that they had a major data breach. And because of that, we have paused uploads from 23andMe, because we can't guarantee that a person is uploading their own data, and not someone else's. So we will eventually get that fixed. I don't know when, but that's on pause right now. There are a few other testing companies out there, such as Living DNA. I believe I've heard of another one, Helix, maybe. We don't accept transfers from them. The tests aren't compatible with ours. So that's why I'm focusing on these. So very last slide here, just some resources. We do have a help center. It's written by this guy, Jim Brewster. So you might want to steer clear of it. These guys kind of wishy-washy. But there are some resources there. There is something called the International Society of Genetic Genealogists, or ISOG. They have a wiki that has a lot of useful terms. If you're on Facebook, they also have a Facebook group where you can ask a lot of questions, and there's a lot of experts there who can help you out. There is a site called Upedia, which if you're like me and you like to nerd out about the sciencey side of DNA, this is a great resource. Unfortunately, it does only focus on European ancestry. If you are aware of a non-European one, I would like to know about it. I have tried finding one, but I can't find a definitive repository of specific information about other parts of the world. But I do find this very interesting, since my own haplogroups are European. There's a blog by a friend of mine, Roberta Estes, called DNA Explained. It also goes into depth with some of these topics. There is GEDmatch. And GEDmatch works like family tree DNA transfers in that regardless of the test company that you've gone with, you can download a copy of your data and upload it to GEDmatch for free. They do have additional features that are behind a paywall, but this allows you to compare with other people regardless of the test company, because each of the matching databases are proprietary and unique to that company. And then lastly is mitoY-DNA, which does the same thing that GEDmatch does, only for mtDNA and Y-DNA testing. So you can upload the haplogroups that you get from the other testing companies. It'll be limited in the information it can give you, but you can upload it there. There are some other companies that do Y-DNA testing but don't do matching, so you can upload those as well. So that's the end of my presentation. Thank you all for bearing with me. And I know, I'm sorry I ended up taking up most of the time, but if you want to go over a little bit, I'm happy to answer any and all questions that you might have.